Hi, today I want to talk about the need for change, to change how we manage disease through interventional technology. Why is it that some diseases are so hard to treat? One of the core reasons is disease heterogeneity. This means that cells in an environment can manifest in many different ways. Now, cancer is one such classic example. In the case of cancer, the survival rate worldwide is only at 67%, and the treatment cost for US alone per year can reach up to $90 billion. But the tide seems to be moving in our favor. We now have a new and powerful tool, liquid biopsy, which can help us manage disease better. This is interesting because we can now obtain clinical samples easier, faster, and at a relatively cheaper cost than conventional methods. In cancer, circulating tumor cells are shed from tumors, and they enter the bloodstream, which can then be extracted. These CTCs, so-called, are extremely heterogeneous, and they seem to reflect the tumor profile better because they are coming from different parts of the tumors and different tumors of the same patient. But these CTCs are extremely frustrating to work with because they are so different from one another and relatively rare compared to the billions of blood cells in the sample. And technologies are not able to fully recover all of the CTC populations, and sometimes they may even compromise viability. So this is why we have the CTC Cluster Assay, which capitalizes on microfluidics and enable us to mimic certain components of the tumor microenvironmental niche. We are now able to generate clusters directly from patient-derived immune cells and cancer cells under hypoxia. What is exciting is not that we can develop clusters from samples of low cancer cell counts, but we can also correlate patient response to treatment and their survival period with cluster forming potential. We are working very closely with the clinicians so that we can bring direct translational relevance for the patients. We have now detected clusters even in early-stage cancer samples, and we are trying to validate this assay for early-stage cancer detection as well as to predict a risk of relapse. What we envision in the future is when we step into the clinics, a patient comes to us and they pass us a few mils of blood. Within 2 weeks, we can provide information that complements other tests and help the clinician to derive the best therapeutic strategy for the patient without undergoing the treatment itself. These are small steps, but we hope that eventually we can make a big change in how we manage disease. I'm Bee Luan Khoo from the Singapore MIT Alliance of Research and Technology. Thank you.